This is our 5th set of notes in Big Idea 1, and I'm going to go as quickly as I can because if you've previewed this, you know there are many slides. Okay, so our connections, we can use math to calculate changes, which is still in Big Idea 1. Distribution of alleles, which is getting to Big Idea 3 within the population. Interactions and homeostatic systems, and we've got all 4 big ideas. Okay, so some things that we need to remember is that individual organisms do not evolve, only populations. Okay, natural selection, however, acts on each individual. This is a picture of a medium ground finch. It went through a drought, and about 90% of the birds were lost due to the loss in the environment of small, soft seeds. Birds that had thicker, stronger beaks and longer survived, and because they could crack the seeds, and they went ahead and passed that on to their offspring. So over time, we ended up seeing that the beak length grew. Okay, when we talk about microevolution, it is the smallest unit of evolution that we can talk about, but we have to talk about natural selection not being the only cause. Up till now, we've just talked natural selection, natural selection, natural selection, but there's also genetic drift and gene flow that can lead to evolution. Okay, so let's take a look at genetic variation. We already know that individuals within a species vary. We can see that just looking around the classroom. Okay, specific characteristics are different for each of us. Some of the characteristics are either something or something else, which is usually determined by a single gene locus or location. So an example of that would be flower color. Some variations exist on this continuum, which results from 2 or more genes or 2 or more loci, and that would be an example of human height. And then some variation even exists just at the gene level, some at the base pair. But usually that doesn't cause a phenotypic variation because we're just altering just one little very minute thing. Another thing that we need to remember with genetic variation is not all phenotype variation is inherited. So an example of that would be a bodybuilder. So humans will lift weights and change their body structures. That doesn't mean that that is inherited. Remember, we're not Lamarck here. Okay. So there are 4 main sources of genetic variation: when we form new alleles, altering gene number position, rapid reproduction, and sexual reproduction. And we have to talk about each one of those. New alleles can arise through mutations or changes in nucleotides. Okay, so is it ATG or did it change to TGT? Only the mutations in a cell that produces gametes or sex cells are going to be passed on to the offspring. Your body cells, those mutations don't get passed on. And that's where most of the mutations actually occur. And usually when we do have a mutation that happens in a body cell, it dies when we die. And then we could also have a point mutation, which is one base change. Sometimes that can have an effect, sometimes it does not. Okay. Any change that can disrupt or take away from the gene can be harmful. Sometimes, however, they can produce positive effects. When a cell is going through meiosis, making sex cells, DNA replication can have slippages or problems where it doesn't copy properly. It can make duplications, things can stick together, and this can allow mutations to accumulate and make new genes. So an example that we can look at is olfactory genes. looking back in history, looking back through our phylogenies, ancestral mammals had about one gene that controlled olfactory or sense of smell. In humans, we now have 350 receptor genes. So over time, small mutations have added up and accumulated. And mice have about 1,000. Another way that we can alter genetic variation is through rapid reproduction. So plants and animals only mutate about 1 per 100,000 genes for each generation. That's actually pretty slow. Sometimes that's even lower or slower in the prokaryotes, the bacteria, but because they have more generations in the same amount of time as we do, their evolution seems faster and we can actually visually see it. So the same would be true of viruses. The example I have for you here is, again, with HIV, and that has a 2-day generation span, which means it's only around for 2 days, and then the next generation is up again. So because of this, mutations are going to happen really quickly. Contrast that with us, who, it takes a minimum of 40 weeks to have another generation. So because mutations can happen very rapidly, that's why people that have HIV, you'll hear them being on drug cocktails, which sounds kind of funny, but it's just basically a bunch of different drugs, hoping that not all of the virus particles will be immune to all of the different drugs at the same time. Okay. Each individual receives a unique combination of alleles from its parents. We know this. Okay. Sexual reproduction is there to shuffle the things that have already happened. Okay, so it doesn't make new things. The sexual reproduction part just shuffles and hands out what was given. So when we do the bean lab and you are taking a black bean or a white bean out, you're just shuffling whatever was in the gene pool. Crossing over, independent assortment, and fertilization can all affect genetic variation when we're talking about sexual reproduction. Okay, now let's get to that Hardy-Weinberg that we've talked about several times. So we know in order for evolution to occur, populations have to have some genetic differences. But genetic difference does not necessarily guarantee that a population will evolve. There are 5 conditions that cause evolution, and we've went over those. And at least one of them needs to be in force. So what Hardy-Weinberg allows us to do is to test whether a population is evolving. So let's review some of the books there. So a population is a group of individuals of the same species that live and breed in the same area. Different populations can be geographically isolated, which means they're somehow removed from the other population, and can then therefore only rarely exchange genetic information because there's something that's physically keeping them apart. In the gene pool, when we speak of that, we're talking about all the copies of every type of allele at every locus, every location, in every member of the population. Sometimes there are things that we call fixed alleles, and that's when we only have one allele for that trait. So, for instance, if all of the dominant alleles were eliminated, like for cleft chin, then nobody would have cleft chin, we'd all be recessive, and that would become a fixed allele. Okay, so here's an example a little bit different than what we've done in class, because our examples were just with simple dominant recessive, and now we have red, white, and pink is going to be our heterozygous. So actually we could use either the homozygous white, which we know our Q is, or we could use our heterozygous because we know that each one of those has both a dominant allele and a recessive allele. Although the white isn't truly recessive. Remember when we're talking incomplete dominance, we have one that is not completely dominant over the other. But regardless, we can calculate. That was my daughter sneezing. Okay, so you can actually calculate the percentages of the P and Q values based on Hardy-Weinberg. Okay. If we need to go over that equation again, just let me know in class. Okay, so the principle behind Hardy-Weinberg and what we're doing is to assess if there is evolution. Okay, that's really why we're looking at this equation. So to determine what the population would be if there were no evolution. So Hardy-Weinberg shows us what that would be. That way we can compare the Hardy-Weinberg to an observed value. And if there are differences, then we know that there's possible evolution. And then we can ask why. What is happening that's causing evolution? The frequency of alleles and genotypes in a population will remain constant from generation to generation if those 5 conditions are met that we'll talk about in just a minute. Okay, so in a Punnett square, when we do a Punnett square, that's going to show possibility in the cross form. And that just shows one possibility. Hardy-Weinberg is there to consider all combinations of all the alleles and all the crosses that could possibly happen in a population, because remember, we're looking at the gene pool. So when we do our Punnett square, we're only looking at mom and dad. But when we actually calculate the Hardy-Weinberg, we're looking at all of the population. Okay, so basically what we can see here is if we place all of the alleles for a certain locus, so a location, in a bin, so they're all in a bin, this is going to represent our gene pool. Well, reproduction occurs by randomly selecting alleles from the bin. An example of this is how fish release sperm and eggs. Okay, so this is assuming random mating. And if we started with 8, 80% of the red allele and 20% of the white, the gametes produced would have an 80% chance of red and a 20% chance of white. So here's a little bit more detailed explanation of the red, white, and pink. Okay, so again, 80% chance of a red allele, 20% chance of a white allele. Okay, so then we get, when those combine, a 64% chance that we'll have a homozygous red individual, a 16% chance that we will have a pink individual, and then only a 4% chance that we're going to have a white individual. So we've gone over the equation before, but here it is again. So if you haven't written it down before, you can. So it's P squared plus 2PQ plus Q squared. So note that the 2's and on the P and the Q should be superscript. The P squared is our expected frequency of the homozygous dominant. The Q squared is the expected frequency of the homozygous recessive. And the 2PQ is the expected frequency of the heterozygous form, whether that's going to be RW, like in the previous example, or big R, little r. So here are those conditions, again, that are necessary for Hardy-Weinberg to be true. And those are, there cannot be any mutations. There assumes random mating, so there can't be any mate selection. Excuse my daughter.
Sorry.
There's no natural selection occurring. There has to be a large population, which is what we're going to try to model in our spreadsheet. So there can't be any genetic drift towards one allele or the other. And then there can't be any gene flow, no movement in and out of the population. Oops. So this is for each individual loci or point on a gene. So here's the thing. One locus can be in equilibrium for Hardy-Weinberg, while another one can't, or another gene could be totally off. So to apply Hardy-Weinberg, we can use this again as the initial test for evolution, and that's going to help us estimate the percent of the population that could have something. For instance, carrying a disease like phenylketonutrients, which are, I'm sure I slaughtered that name, but they are the people that cannot process phenylalanine. So you'll see on the can of Diet Coke or of a lot of different products contains phenylalanine. They have an issue with an enzyme that doesn't allow them to be able to process that. Okay, so this only happens in about 1 in every 10,000 births. If we apply the Hardy-Weinberg principle to it, we would expect that about 2% of the population would end up with this PKU, and that's actually what we do see because there is not much evolving or changing happening in the U.S. population. We have good medical care, and we can monitor our diets to make sure that we're not taking that phenylalanine in if you happen to have a problem with it. But in other countries, they might not have the medical care or the knowledge that they need, and that allele could be almost eliminated from the gene pool because the people don't survive long enough to reproduce. Okay, so now let's talk about how Hardy-Weinberg can be violated, and that didn't sound good, and how of violations of the Hardy-Weinberg conditions and what that means for evolution. So the first two conditions, that doesn't really apply to us. So no mutations and mating don't really affect the allele frequencies that much. So we focus on the last three. Okay. Natural selection we've talked a lot about, but by consistently favoring some alleles over the other, nature can cause what is known as adaptive evolution. The better the match between the organisms and their environment, the better the organism is able to survive and reproduce and pass on its traits. So an example that I have here are fruit flies and DDT resistance. A long time ago, before we started using DDT, the fruit flies weren't resistant to it. Well, we started using it. Some of them had a mutation that allowed them to be able to survive. And now we see many different strains of fruit flies that are actually resistant to the DDTs. Genetic drift is another thing that can cause evolution. The smaller the population, the more likely the chance that a deviation will occur from what we're expecting. So, for instance, if you flip a coin and you flip the coin 10 times, if you got 7 heads and 3 tails, you'd probably just shrug your shoulders and say, oh well, and then you'd maybe try again. However, if you flipped a coin 1,000 times and you got 700 heads and 300 tails, you would maybe check out that coin, figure out what was going on, because that wouldn't be what you would expect to get. You would expect to get about 500 and 500, give or take a few, but not 200. Okay? So genetic drift can actually work in the same way. So the smaller the population, the more skew we can see towards one way or the other. So here's an example of genetic drift. If we look at generation 1, we started and there were 5 red flowers, 4 pink ones, and 1 white one. So the P frequency or the R allele was 70%, and the frequency of white was only at 30%. So don't forget we have to count the pink ones, that they have a white allele also. So if we're only looking at the ones that are highlighted in white, in the white boxes, then we're going to say that those are the ones and the only ones that could leave offspring behind. So then we come over to our second generation, and because there was only one red one over here that left offspring, and 3 pink and a white, now this is what we get. We get 1, 2, 3, 4 pinks, 3 whites, and 3 reds. Okay. Now we're going to say that we've got some more organisms leaving the gene pool, and only 2 of these plants are actually going to leave offspring, and they're both the red ones. And now we've completely eliminated any other allele except for the red. There are different effects that end up happening because of evolution and because of change in populations. And those are some things that we should go over, one of which is the founder effect. So if a storm were to carry some new individuals to a new island, which can happen, it carries a bird off its path and into a new area, then, and if that population is now isolated from the old population, they will establish a different population that will eventually have a different gene pool from their original source. So that would be the founder effect. Another one is the bottleneck effect. The bottleneck effect usually happens when there's a sudden change in the environment. And for some reason, whatever that sudden change is, it drastically reduces the numbers of the population. Just by chance, because of that drastic reduction, certain alleles may be either overrepresented, underrepresented, or not represented at all, like meaning completely lost. So what this does, this bottleneck effect, it can maintain low levels of variation for long periods of time. And unfortunately, humans are the ones who have caused a lot of bottlenecks for many other species. So a little graphic example of what's happening here. So if we have our original population, you can see blues, whites, and a few yellows mixed in there. Well, literally creating a bottlenecking event that only allows certain species through, and therefore only certain alleles through, we end up with a surviving population, very small, of just blue and yellow. So the blue alleles are overrepresented, yellow is underrepresented, and white is underrepresented, and yellow is completely gone. This happens with the Florida panther. We have eliminated a lot of their species, and their genetic pool has shrunk, and some genes are overrepresented and some are underrepresented. This also happened with the Illinois greater prairie chicken, where we took away a lot of their environment, and so they went through a bottleneck, and you can see by the green area where all of the prairie chickens were before the bottleneck, and because we took away all their habitats, now they only have these 2 little areas back in 1993. So the number of the population decreased, and therefore the number of alleles that were available actually decreased along with it. And it had to do with the percentage of eggs that the chicken was able to hatch. Okay, so as a summary, genetic drift works mostly on small populations, or it's considerable on small populations. It can cause allele frequencies to change at random, so it doesn't necessarily have to be one force or another. It's just a random situation because of the small populations. It can definitely lead to a loss of genetic variation and can cause some alleles to become fixed, where that's the only allele available for that locus. Okay, and gene flow is that third one that we're going to talk about that affects evolution, and that is the transfer of alleles into or out of a population. So emigration and immigration. And that's only for fertile individuals. We don't care if organisms move in or out that aren't fertile because they're not going to affect the gene pool that gets passed on. What this does is reduces the difference between populations because we have a flow back and forth of genes, and sometimes it can affect how well an organism can be adapted to its environment. How well the species can be adapted to its environment. Okay, a lot of times you hear the terms survival of the fittest or struggle for existence associated with natural selection, and Darwin. But those really are misleading terms because they're basically saying there's direct competition between individuals. Well, you're more fit than this one, so you will survive. Okay? It's really kind of a lot more subtle than that. So we can look at it in terms of relative fitness. And that is, like we've been saying, certain traits can lead to greater contributions to the gene pool or the greater ability to survive and reproduce. So it really is kind of survival of the most fit to that environment at that time. But that's too long to say. Okay, there are 3 types of natural selection that we need to talk about. And we'll go over what those graphs look like in a minute. And those are directional, disruptive, and stabilizing. And this is going to depend on which phenotype or physical look is favored in the environment. So directional selection is going to occur when the conditions will favor individuals at one extreme of a phenotypic range. Okay. So really tall, really bright feathers, really big plumage. So it would shift everything in one direction or the other direction. Directional selection is very common when there's environmental changes or when migration occurs because we're losing some of that gene pool. Okay, disruptive selection occurs when individuals at both extremes of the phenotype are favored over that heterozygote intermediate form. And that's going to shift the curve both to the right and left at the same time. So we're going to select for tall and short and not medium height. Okay, and then finally there's stabilizing selection, and that favors the intermediate phenotype and acts against both of the extremes. So it's going to favor that heterozygous condition, that intermediate. So here's some of the graphs, and I think I have it on the next slide also. Nope, I don't. Okay, so let's take a look at these. So here's our original population. Okay, so if we have a directional selection, it's going to favor one of those traits that's going to pull it either to the left or to the right. The disruptive selection is going to favor the alleles here and also the phenotype for the alleles here, but not so much in the middle. And then finally, stabilizing is going to do exactly what the original did, and that's just going to go right along the same curve and favor the intermediates. Now, there are forms of sexual selection where certain inherited characteristics are going to make an organism more likely to obtain mates. We've seen this in a lot of different circumstances. It can result in a situation called sexual dimorphism, where we have differences in sizes and colors, in ornamentation and behaviors between the two sexes. Now we get to talk about competition and selection between and within the sexes. So intrasexual selection is when the same sex individuals compete for the opposite sex. So, ladies, an example of this would be when the girls dress up for each other. You're trying to look better than the girl next to you so that you can get the guy. So you're both competing for that same guy. Or guys, same thing. You're trying to look better than the guy next to you so that you can get the girl. Okay. And then intersexual selection is mate choice. This is where, and it's usually the females, sorry guys, that have the choice, and they are very choosy when they're selecting their mates. So they only want the males with the biggest plumes or the brightest feathers. So sometimes this can actually put males at risk because of showiness. They're going to maybe have structures that might not be very good in being able to escape predators, or might make them a tag saying, hey, here I am. But it's good because they get selected for by the girl. So here's a male and female peacock. So a peahen and a peacock. Okay. And he's showing her his beautiful plumage, and I don't know, maybe she's being receptive, maybe not. But she's actually doing mate selection. So that would be intersexual selection. And then finally, how do we preserve that variation when we're talking about natural selection? We can have something called neutral variation, which are differences in the DNA that do not give a selective advantage or disadvantage, either way. That usually comes because organisms are diploid or have diploidy, which means that they have a lot of hidden recessive alleles, like with the cleft chin that we've talked about, or sickle cell anemia, where you have big S, little s, and there's a lot of those little s's hidden in there. Balancing selection will help to maintain 2 or more forms in a population. Okay, and there's 2 main types of those: heterozygous advantage and frequency dependent selection. So in heterozygous advantage, obviously individuals that are heterozygous have greater fitness than both the homozygotes, and that's where that sickle cell comes in. People that are big S, they don't have sickle cell anemia, but they are susceptible to malaria because of the shape of their blood cells. People that are little s, have sickle cell anemia, and most will die because of not being able to receive proper medical treatment. But in the heterozygous form, they have the advantage because they don't show sickle cell anemia, but they also don't get malaria. So that's the heterozygous advantage. And then frequency dependent selection is where our fitness will depend on how common it is in the population, and usually will change. And the frequency will depend from year to year on what was most useful. Okay, so if something is a left-mouthed fish, it's not going to be able to attack its prey from on the left side. It can only attack it on its right side. Okay, so usually in this case, what we'd see is kind of a back and forth every year as to who survives and who doesn't. So why hasn't evolution and natural selection produced any perfect organisms? Well, because selection can only act on existing variations, not it doesn't create new ones. Okay. we don't have a perfect organism. Also, evolution is limited by its legacy from the ancestral form, so it has to come from somewhere, and then we make changes or build upon those legacy, those historical ancestral forms. And then a lot of adaptations, we have to remember, are often compromises. A great example of that is humans have these amazingly flexible limbs, which help us do a lot of things, but there's a compromise that comes with that because there could be a lot of injuries that are sustained to our limbs because of their flexibility and limberness. And then chance, natural selection, and the environment are also interacting, causing organisms problems. So natural selection really just operates on a better than basis. Having this is better than this at this point in time in this environment. Hopefully we got all that, and please ask questions if you don't understand. Thanks. Good night.